All set? Welcome everybody. My name's Mark Rosenthal. I'm a medical oncologist and I'm the VCCC Clinical Trials lead, running a clinical trials program. Lots of things that we're doing to try and improve access and participation for clinical trials, and as I'll come to, delighted to introduce Peter Gibbs in a moment. As we always do, we acknowledge the traditional owners of the land in which this event is taking place, the land of the Wurundjeri people of the Kulin Nation, and pay respect to the elders and families past and present. So you're aware that the VCCC is a large alliance of medical research institutes and hospitals. Peter has a number of positions, as you can see. He's the GI Education Research Lead for the VCCC. He's a laboratory head at the WEHI, and he's a medical oncologist at Western Hospital. Peter, I know how hard he's worked to get to this point. He sat in an office one down from me over at Royal Melbourne for about 15 years, never heard a word out of him, just typing away at his desk, got there before me, left afterwards, and he's been incredibly successful over many years. What he's going to talk to us about today is turbocharging clinical trials, biomarker and registry randomised clinical trials. So I might just say that here I am, the VCCC clinical trials lead, and I'd never heard of registry trials up until about 8 months ago. So I'm supposed to be an expert in clinical trials. I'd never even heard of it. Peter's an expert, and you're going to hear why this is so exciting. Thanks, Peter.
Good afternoon. Thanks, Mark, for the introduction. So one of the key goals of the VCCC is to increase clinical trial accruals. And I'm talking today about two slightly different approaches to clinical trials that I think will help us to meet some of those goals that we're setting, particularly biomarker studies, which we've started, but looking to rapidly expand and moving into randomized studies, and also some registry clinical trials that we're about to start. This was a presentation at ASCO last year. It was a nice summary of the history of clinical trials, going back to 1747 with the first randomized study in 12 patients, 6-arm study proving that vitamin C prevented scurvy. Things have changed a lot since then, as shown in the timeline along the bottom, but randomized clinical trials have predominantly stayed the same, with a particular emphasis on ethics in the last number of decades. As a medical oncologist, we have a lot of challenges in the clinic, and one of the challenges is the success that we've had, and that creates options for our patients, which is terrific. We're seeing better outcomes, but we have to be able to choose the best option for the right patient at the right time. And as we get more options, those questions, those considerations become more complex, for medications, the biologics, other interventions, including surgery and radiotherapy. So a number of dilemmas about what is the best treatment for the patient that sits in front of us. And we're also understanding that for each cancer, there's many different types of subsets. We don't just have colorectal cancer anymore. We have a left-sided and a right-sided colorectal cancer, and many molecular subtypes that are being identified as well. So I've got lots and lots of options and an increasing number of subsets. So a major challenge: how do we move forwards? How do we provide the best treatment for our patients? Traditionally, the focus of clinical trials has been on developing new drugs and new drug combinations, and we compare those to a current standard, typically in a randomized phase 3 study. We don't, unfortunately, do as much work on defining the optimal duration, sequencing, and combinations, which is increasingly needed as we develop more active therapies. And we also haven't paid a lot of attention, probably a lot less than we needed to surgery, radiotherapy, palliative care, and other aspects of cancer therapy that are just as important sometimes as the drugs that we give to our patients. And in the category at the bottom, rarely do we look at biomarkers, which I think are very important in helping us to define which treatment to use in which patients. But most of these have only been defined in post-hoc analysis and rarely have been integrated into standard care. So as a summary, I think there's an ever-increasing gap between the outcomes that potentially could be achieved if we delivered optimized care. That's the right drug for the right patient in the right sequence versus what we're doing currently. And along with this, there's an exponential increase in the number of questions that need to be asked in randomized controlled trials. So I'm talking today about biomarker-driven randomized controlled trials and giving an example with circulating tumour DNA, and also about registry randomized controlled trials. And some of the attractions of these are that we can ask more questions more quickly. They're relatively simple and cheap, and they can be widely engaged. So hospitals that don't have particular resources in terms of fancy imaging, research nurses support, etc., can still do these studies. So to talk about circulating tumour DNA and the way that we might be using that to improve treatment of our patients, and as background, as a medical oncologist, part of the MDT, our patient presents with cancer, such as bowel cancer. We do scans to make sure there's no spread of the cancer, and then the surgeon usually goes ahead and resects the primary tumour. We as a group look at the pathology from that cancer specimen, try and work out what the risk of recurrence is, and decide whether or not to give some adjuvant therapy, some additional drug treatment for that patient for a defined period, usually 3 months or 6 months, and then we wait and see. Unfortunately, with this approach, some patients get treatment that don't need it, and some that don't get treatment that do need it do get it. So one of the concerns is that patients will still have a recurrence despite our best intentions. And a sensitive and specific marker of residual disease that defines these patients likely to recur could significantly improve our treatment of these patients. So as an introduction, circulating tumour DNA, there's small fragments of DNA that occurs and is released from the tumour. These are mutated and different to the normal DNA that is present in the circulation. So we've got a mutated DNA that's quite cancer specific, and if we can measure that in the blood, it's an indicator that somewhere in the body there's a tumour, which may be very small, that's releasing the circulating tumour DNA, and that's something we can measure and potentially use in the clinic. There's a broad range of applications of circulating tumour DNA, right from screening through to treatment of advanced cancer. But today I'm focusing on what I'm calling minimal residual disease, trying to define those patients that look like they've been cured by their surgery, but are at high risk of developing recurrence unless they receive further treatment. A very good example of one of the dilemmas that we have in the clinic looking after colorectal cancer is stage 2 colon cancer. And it's a bit of the medical oncology lottery. So 80% are cured by surgery, which obviously means 20% are not. And over the last 20 to 30 years, we've done lots and lots of trials working out, trying to work out how better to treat these patients. But typically, if we have this case, 50-year-old male with a T3N0 cancer, stage 2 cancer, a high-risk feature, walks in to see a medical oncologist. Anywhere in the world, he might receive no treatment, he might receive 5-FU, he might receive 5-FU and oxaliplatin. If he's treated, he might be treated for 3 months or 6 months. And if he comes in next month, he might see the same doctor and get a different treatment. We don't really know what the best treatment is. All of these things might be helpful, but it creates a distressing situation for the doctor. Do I treat the patient or not? If I don't treat them and they recur, I'll feel bad about that. If I do treat them and they get side effects, I might feel bad about that as well. And it's also very confusing for the patients. So what we've tried to do in the past is to improve outcomes in stage 2 colon cancer and other diseases by adding therapy. We've added 5-FU, 5-FU and oxaliplatin, 5-FU oxaliplatin bevacizumab over a series of decades with clinical trials. Some, like the one shown here in stage 2, the blue curve versus the yellow curve, a slight difference in terms of recurrence, but really quite a confusing picture about what to do for these patients. Another traditional approach that hasn't really worked to date is looking at tumour biomarkers, trying to work out if there's features of the cancer that tell us more about who's likely to recur and who isn't, and to use that then to decide who to give chemotherapy to. And shown here is an example of one of these expression-based recurrence scores. It does divide patients into different risk groups, but the difference between the risk groups is pretty marginal, and a hazard ratio of 1.38 I think is quite unimpressive. So on the left is the traditional way of assessing patients. The tumour's removed, we look at the tumour under the microscope, estimate the risk of recurrence, and then decide whether or not to treat the patient to try and reduce the risk of the cancer recurring. An alternative approach based on circulating tumour DNA could be to take a blood sample after the surgery and see if there's any circulating tumour DNA in the blood. That would then be a direct evidence that this patient has some residual cancer somewhere in the body that's releasing that mutant DNA and tells us they've got very high risk of recurrence. So that's the theory. What about the evidence? So this is the first trial that we did in patients with stage 2 colon cancer, had the surgery removed, looked to be cancer-free. We took their tumour tissue, we found a mutation in their cancer, and then we went looking for that same mutated DNA in their bloodstream. And then we followed the patients up to see what happened. So clearly this does predict recurrence risk. So this is the 3-year recurrence risk of patients according to their DNA status. So if they had DNA detectable in their blood, 100% of patients recurred. If they didn't have DNA in their blood, 92% of patients didn't recur. So it nicely divides up patients into high risk, very high risk, and very low risk groups, which is very important for a biomarker. And just to emphasize, in the multivariate analysis, the hazard ratio was 28, which is quite impressive compared to 1.3 or 1.4, which some of the other biomarkers have produced. We've done a series of studies over time looking at different types of cancer, particularly GI cancer, and we've seen a very similar picture for circulating tumour DNA across multiple disease contexts. So for rectal cancer, if DNA is detectable on the left in those that have had a great pathologic response, or on the right for those that have had a poor pathologic response, it still very nicely divides up into very high risk and very low risk cancers. This is another study with resectable colorectal liver metastases, again dividing up into 2 very clear groups, high risk and low risk. And also one of the consistent features of these studies is that if we do detect DNA and patients don't have chemotherapy, recurrence is close to 100%. So clearly that defines a group that needs further therapy if we're trying to avoid cancer recurrence. Probably the most exciting data we've seen so far is the recent data in stage 3 colon cancer, which we've just started to look at. So as I've said, if you've got circulating DNA in the blood, that predicts somewhere in the body as residual cancer and close to 100% risk of recurrence. We do know for stage 3 colon cancer, unlike stage 2, that this group of patients with involved lymph bands do benefit from adjuvant chemotherapy. It reduces the risk of recurrence by maybe 30 or 40%. And what we've seen in our initial analysis of our stage 3 cohort is that we can measure the benefit of adjuvant chemotherapy rather than saying this patient is node positive, we should treat them for 6 months and see what happens. We can define who's at very high risk, and we can actually follow those patients through their treatment to see whether the treatment's working or not in that individual patients. So if patients start with a positive DNA after their surgery, becomes negative on chemotherapy, tells us the chemotherapy is working, the risk of recurrence is 5-fold or less smaller. And if we do see during chemotherapy that they go from negative to positive, their recurrence risk is extremely high. So it's quite exciting to see circulating DNA as the first ever real-time marker of adjuvant therapy in impact in solid tumours. So potentially could hugely improve our treatment selection and our monitoring of whether the treatment is being successful or not. Obviously, to validate a biomarker, there's a final step, and that's proving clinical utility. So what we need to do now is multiple randomized studies looking at using DNA as a biomarker to stratify patients and then to follow to see if that's having an impact, and hopefully ultimately proving that we're curing more patients with this biomarker-informed approach. So this could be a number of different trial designs. It could be like our stage 2 colon cancers, where standard of care maybe to treat, maybe not. And those that have a positive DNA, we give them chemotherapy, and negative DNA, we decide not to. That's a pretty straightforward trial design. It gets a bit more complex when the standard of care is treatment. But one of the possibilities here, as well as ramping up, giving more aggressive treatment to those that need it, is also de-escalating or giving less treatment or no treatment to those that don't need it. So having a high-risk group that we treat more aggressively and a low-risk group that we can back off from and hopefully give them less treatment and less side effects. So that's ctDNA-informed or biomarker-informed clinical trials, and potentially we could be looking at ctDNA studies across other disease types, but also other biomarker-informed studies could be quite helpful in terms of moving our treatments forward for colorectal cancer and many other tumour types. So the rest of the talk I'm focusing on registry-based randomised controlled trials, and this is supported by significant funding from BCCC, and hopefully is one of those programs that will increase clinical trial recruitment, not just across the BCCC sites, but also across Victoria. So I'm going to overview real-world data and real-world registries and give an introduction to what has been done so far, and then talk about registry randomised trials, particularly in medical oncology, and how they could be useful to move the field forward. So as we know, prospective randomised controlled trials remain the gold standard for defining safety and efficacy of new therapies. So each time we want to make an improvement in treating patients with cancer, ideally we'd randomise patients as part of a controlled trial and demonstrate that the new therapy is better than the old therapy, more efficacious, and hopefully also safer. But one of the interesting things when you look at randomized controlled trials in colorectal cancer and many other tumor types is the randomized trials, even when they demonstrate a benefit for the new therapy, don't impact on routine care. And to give an example from, again, the context of colorectal cancer, for patients with metastatic colorectal cancer, people that walk in and unfortunately the tumour's already spread to the liver or the lungs, in most places in Australia, you'll be given a chemotherapy, oxaliplatin-based chemotherapy called Folfox. So that's about 90% of the patients. So we collect data in our registries and we can see what the standard of care is. About 90% of patients will get Folfox. That's despite 4 randomised studies that have compared single agent 5-FU, so a lesser treatment that has less toxicity, to our standard of Folfox. So 4 randomised studies have shown that the single agent's as good as the Folfox in terms of overall survival, but it's not widely adopted. And there's also a triplet approach using all 3 chemotherapy drugs to treat metastatic colorectal cancer, a randomised study showing that improves survival by 5 months, but also minimally used. If we look at the biologics that we use to treat metastatic colorectal cancer, a classic biologic that we add to Folfox is bevacizumab, and again, based on our registry data, about 80% of patients will receive bevacizumab. So the chances are if you walk in to see a medical oncologist, they'll offer you Folfox and bevacizumab as treatment for your metastatic colorectal cancer. Now that's despite data again showing that there's alternative approaches, such as using an EGFR inhibitor, that actually make it better or create a better survival outcome, but it hasn't been widely adopted. So just to summarise, there's lots of clinical trials that have conducted, but they haven't changed practice. There's lots of reasons why that is the case, but one of the potential concerns often of medical oncologists is what we call limited external validity. So what that means is that the trial results may not necessarily translate to the patient that sits across from the desk in the outpatient clinic. So trials are very selected. They're fit in young patients. They're conducted in highly specialised centres according to strict protocols. The questions are, can the efficacy data be reproduced? Do we get the same survival benefit from adding a study in a 75-year-old patient who may have other significant comorbidities? Is it safe to treat an older, frailer patient? And are the biomarkers valid? So our left versus right analysis for colorectal cancer is a valid way of deciding which patients to treat. And unfortunately, as mentioned before, most of the biomarkers that we use have only been looked at post-hoc. People have conducted a clinical trial. They've looked at the patients with the left side or the right side of cancer and tried to work out what is the optimal approach in that group of patients. But really, to be confident, you need to do a prospective study of left-sided colorectal cancer and right-sided colorectal cancer. So to summarise, clinical trials, the standard clinical trials that we do are conducted in ideal circumstances, typically in high-volume hospitals. So not so much in the smaller hospitals, rural centres, private hospitals. They tend to be big public hospitals that have specialist doctors, and that could be a GI specialist that just sees colorectal cancer, nurses that are specialists in treating cancer patients, expert pharmacy, expert radiology to interpret the imaging. So it's an idealised environment for running a clinical trial looking at the impact of a new therapy. The subjects are highly selected and highly motivated. So they're likely to be compliant with their therapies, particularly oral therapies, likely to turn up for their visits, likely to report their side effects, and likely to follow advice. But that may not be the case when we look at the patients that we see in routine care. And the treatments that are delivered in clinical trials are also according to strict protocols. And there's very little margin for movement from that strict protocol. You have to follow that or you get into trouble. So there's this concept of treating patients in randomised controlled trials, demonstrating a benefit, and then hopefully what will happen is that that new treatment, new approach will be taken up into routine care, and then we'll see the benefit in the patients in routine care reproducing what was seen in the clinical trial. As I've said, there's a major concern about the selective nature of trials and whether the data may be translatable to routine care, and that's leading a major interest in what's called real-world data. So we can't conduct the trials necessarily in that real-world population, but what we can do after the trial is finished is collect data in the context of routine care, look at what the treatments are that are being implemented and see what the benefits are, and hopefully get some evidence that the benefit that was seen in the randomised controlled trial is translating to benefits in the patients. So the definitions of real-world data is any data that's collected on patients from the hospital record or into a registry outside of a standard clinical trial, and that creates real-world evidence, which is the evidence that's derived from analysis of the real-world data. So typically with medical records 10, 20 years ago, used to all be paper-based, used to be down in the dungeon of the hospital, and used to be difficult to access. So we've moved quite quickly now to electronic records, paperless systems, where all the data is captured in a uniform way and is accessible electronically from anywhere in the hospital or even outside the hospital. So it's made the data much more accessible for research purposes as well as for routine patient management. And it's also meant that we're capturing data in electronic form rather than a written form. So this is a chemotherapy prescription on the right from Western Hospital, written by one of my colleagues. Very difficult to decipher, potentially quite dangerous, but it's hard to actually go back into the medical file and pull out the data that you're after when you have to try and read the doctor's handwriting. So if data is captured in electronic format, it's very easy to interpret. So now we're getting lots of data that's being captured as part of this real-world effort into electronic records. But we can't necessarily presume that the data that we're after is included in the hospital file. This is a paper from about 9 years ago, but I think quite relevant still to the current context. In the US, a group of researchers were going through hospital data and looking at treatment of metastatic colorectal cancer. And on the top there, they were showing that the diagnosis of colon versus rectal cancer, that can be quite difficult, so not too fast if we can only see 14%. But 27% couldn't tell the stage of the cancer, and most extraordinarily, 17%, they couldn't actually determine the sex of the patient. So very basic data that you presume, you just flick through the medical record, it's there, you can extract it for research purposes, isn't always collected and not collected reliably. So one of the things that I've been looking at probably the last 15 years is registry data collection, and doing a little bit different to what would be considered the standard registry. So when people talk about registries, typically what they're talking about is someone going back through medical records, maybe 6 months, 12 months after the event, trying to extract data and collect it into a registry. What we've been trying to do is to focus more on what I would call real-time tumour registry data. So that's combining the information that's in the hospital record, but also asking particular questions around data points that aren't well captured as part of the standard medical record. So we've got the data from the electronic health record, but also a registry database that captures additional important information. So this type of information that's critical to understanding patient treatment and outcomes is shown on this example here. So we've got a drop-down box at the top for ECOG performance data. So that's a measure of the patient's general fitness. It's a major determinant of outcome, but it's not reliably captured in the medical record. If you go through most medical records, it'll be documented in less than 10% of patients. Another thing that's not well documented is the comorbidities and also the family history. So by creating these registry data sets and databases, we can reliably capture, hopefully on every patient, their ECOG performance status, their comorbidities and their family history in a standardized way, a drop-down box or a tick box format. So all of the information is there and it's reliably collected. So it can be used to interpret that patient's condition and their treatments and outcomes. So as an example of one of these registries, the first major one that we set up, the TRACK registry from 2009, a consensus data set for metastatic colorectal cancer, collecting data across all lines of therapy at multiple centres across Australia, now internationally, and collecting data on consecutive patients, trying to standardise the data that's collected and maximise our opportunities to use that data for research. But there are major concerns with registry data, and just to highlight a few of those, you can't necessarily look straight at the registry data and be confident that what you're seeing really makes sense. So to give an example, this is an analysis we did looking at the time to start chemotherapy. So if there's major delays in starting chemotherapy for our patients, you'd presume that's a bad thing and would lead to worse outcomes. In this example, however, the patients that started chemotherapy quickly within 30 days had a worse survival than those that started chemotherapy after 30 days. And if you think about it and you understand the disease, you can understand that the patients that start chemotherapy slowly are those that may have quite low bulk disease or may be going to have a liver resection, which is a very good prognosis group. The patients that start chemotherapy quickly are those that are likely or destined to do quite badly. So you're comparing apples and oranges. And many of the retrospective registry-based analyses are these apples versus oranges comparisons. And you might be able to understand it when you look at this type of comparison, but many other times it can be difficult to work out what's the truth and what's not the truth. So another example looking at registry data and how it can potentially be misleading is from, this is analysis of a registry in the US. And this was when Avastin, bevacizumab, was first approved for metastatic colorectal cancer. And it was shown adding bevacizumab to chemotherapy in the first line setting as part of initial treatment led to a major improvement in overall survival. They collected registry data on the use of bevacizumab in a clinical, in a hospital population, and then looked back to see what the impact was when bevacizumab was used in the second line setting. And what they found in this analysis was there was about a 10-month, 11-month improvement in survival when bevacizumab was added to chemotherapy. So a very dramatic difference from this registry analysis. However, as I mentioned, there's many biases and confounding factors that determine which patients do or do not get chemotherapy. And in response to that publication, there was this letter written suggesting that this may be the case. And they performed an analysis of patients who had ondansetron, an anti-nausea drug that was used for chemotherapy or not, and showed that if you had ondansetron as part of your therapy, you had a much better survival than if you hadn't. That's obviously totally independent of the therapy received, but makes the point. So more importantly, ultimately, and like we always need to do to be confident about results, a prospective randomized controlled trial was conducted. So the registry analysis suggested a 10-11-month survival benefit. The randomized clinical trial suggested a 1.4-month difference in survival. So something, but not very much compared to what was seen in the registry analysis. So how can we take registry data and move to registry randomized controlled trials? So the VCCC is supporting this across multiple tumour types and across multiple disciplines. So one of the benefits, hopefully, of registry trials that we can answer not only chemotherapy questions, but also questions regarding surgery, radiotherapy, and palliative care. So there's a number of questions that potentially could be asked in a registry trial and a number of questions that can't be asked in a registry trial. And just to illustrate that, for pancreatic cancer, we have two standards for our first-line treatment. Our old standard was gemcitabine, a single-agent treatment. A prospective randomized controlled trial demonstrated that FOLFIRINOX was better than gemcitabine. Separately, another clinical trial or series of trials demonstrated an alternative, nab-paclitaxel gemcitabine, was better than gemcitabine. So we've now got two studies in the first line showing benefit compared to gemcitabine. So a critical question is FOLFIRINOX or gemabraxane our best first-line therapy? And doing a head-to-head study of those two would be one way of defining which is the best treatment. Unfortunately, that wouldn't be possible as part of a registry trial. So the funding environment can be important, so that drugs may not be available in the context that you want to use them, but also clinician equipoise. And what I mean by that is some clinicians are convinced that FOLFIRINOX is better, some are convinced that gemabraxane is better, and if you opened a trial and clinicians didn't recruit to the study, that's not going to be potentially very useful. So talking about, or going back to the start, talking about the current trial landscape, that's a pretty classical clinical trial comparing two new therapies for metastatic pancreatic cancer. But moving on to this concept of what is the best way to sequence and combine our therapies potentially could be looked at in the context of a registry trial. So one example that we've put together for a registry trial is a study in glioblastoma. So we have data that's being collected in a glioblastoma registry. It's collecting data on consecutive patients. We collect data on the treatments that people have, the outcomes from those treatments, and survival. So all that information is relevant to a clinical trial. And the only extra thing that we're doing in this study is randomizing patients to two different therapies and then capturing the data in the registry. So as background for this study, there's an emerging standard in the US where 12 months of therapy, rather than the standard 6 months of therapy, is being adopted largely based on case series. So we have a question, is 12 months of temozolomide better than 6 months of temozolomide? There's no randomized controlled trials presented. As far as we know, no randomized trials underway, but it's an important question. And the danger is if this is used for long enough, it becomes a de facto standard without the evidence there to support its use. So this would be patients with newly diagnosed glioblastoma. They complete their standard initial therapy of chemoradiation and temozolomide, and then after that's completed, are randomized to observation, so to receive 6 months of treatment only, or an additional 6 months of temozolomide, and then we follow them with the registry to look at overall survival as our endpoint. So this incorporates, the registry randomized control incorporates the standard and important critical elements of traditional trials. So we're identifying a patient group, a standardized patient group. Patients are consented for the trial, and hopefully some biomarker studies as well. We stratify according to known prognostic factors, and then critically we randomize to either 6 months of standard or to 12 months of temozolomide. And then, as we would in any other trial, we treat, we follow up, and we collect data. Importantly, for these trial endpoints, for registry trials, what we're trying to do is collect hard data endpoints. So overall survival, patients, unfortunately, they're alive or dead. It's very clear cut. We're not trying to collect information that may be more difficult to interpret. And to support these analysis of overall survival, we'll be linking data with the death data from the Victorian Cancer Registry. And all of the data for the trial, including the death data, is collected in the registry. So I mentioned we've got the brain registry. It's collecting the data on each of these patients as they come through and used for a whole lot of purposes. And for the registry randomized trial, all that we're doing is randomizing to one of two standard of therapies and then following patients through to try and answer important questions. This could potentially be adopted, the registry trial, in many other contexts as well. This is an example of a study in palliative care that we've been looking at potentially for pancreatic cancer. So a previous study in lung cancer demonstrated that early referral to palliative care in people with advanced lung cancer compared to standard of care referral later on at the time of symptoms or other need led to a major improvement in overall survival. This is one of those studies where we've got a prospective randomized study, but clinicians are a little bit unsure about whether or not that makes a difference. I'm not sure whether, based on this study, all the lung oncologists are now referring the patients for early palliative care. It's an important study, potentially, but not sure whether it translates to the Australian context and to the broader clinical population. So we thought we could ask a similar question in pancreatic cancer. So these are newly diagnosed pancreatic cancer patients. They're having their data entered into the purple registry. And what we're doing is to stratify according to known prognostic factors and then randomise to our standard of care management, chemotherapy, etc., or to early palliative care. So the only intervention, the only thing we're doing differently is to consent patients and refer early for palliative care. We're following them through the clinical course and looking at overall survival as our endpoint to try and answer this question of early palliative care referral in pancreatic cancer. So importantly for registry trials, they're not relevant or applicable to all contexts or all questions. And really what we're trying to do is compare different standards of care, and particularly for that pancreatic example, where there's two randomized studies that have demonstrated new approaches are better than the standard of gemcitabine. Obviously, you have to have access to the intervention. So we've got palliative care for the palliative care study in pancreatic cancer. For the glioblastoma study, we've got access to an extra 6 months of temozolomide. They're available interventions. We've got hard endpoints for these two studies, overall survival, but other studies could have different endpoints, and potentially we could have different models for consent, and hopefully look at research nurses or other ways of consenting patients, so it makes it less likely that the doctor could forget about the study or forget to consent the patient. So trying to optimise study recruitment. And as part of these studies also to routinely consent for translational research, so to get prospective consent for biomarker-based studies that could add value in terms of understanding which groups benefit most or least from the new intervention. So for our registry trials, what we're doing, and to contrast that with the standard clinical trials, which compare a new therapy with an old in ideal circumstances, these will be what we'd call usual circumstances, and hopefully be adopted beyond the high volume centres. So there's no reason why a smaller private hospital or even an individual clinician couldn't participate in these studies. There's variable specialisation and research expertise. It's an unselected patient population versus a very carefully selected population. What we're really trying to do is to work out whether these new interventions, these alternative ways of treating patients, can improve real-world outcomes. There should be also some flexibility with the regimen. So rather than a tightly fixed, protocolised driven way, there should be flexibility to manage patients according to standards of care. So this is the metastatic colorectal cancer registry, not quite up to date, but until recently we had 25 sites across Australia collecting data. There's also a number of international sites. So these registries can be quite widespread and can be collecting hundreds, if not thousands of patients. So very large numbers of patients that are available for these registry trials. Also in green, the private centres, so data is being collected in private as well as the public hospitals, and these studies could be run in private. And also there's a number of smaller regional centres that are also participating in these registries, which means all of these settings can contribute to recruitment to these registries.
Floor, volunteers department, attention, code red, ground floor, volunteers department, and drill code labor.
All of these centres can contribute to these registry trials, unlike increasingly with some of the industry-sponsored studies being very selected, selecting only 1 or 2 sites in Melbourne or 1 or 2 sites in Victoria that can open the study. To summarise there. So just to summarise the conclusion, the VCCC set an ambitious goal of increasing clinical trial activity, and to be achievable, I think we need to be looking at our standard way of doing things, but also different ways of doing studies. Biomarker studies, registry trials is one way of meeting these targets. Hopefully I've convinced you that biomarker studies and registries trials are valid alternative strategies that can support studies that are potentially practice changing, improve the treatments for the patients, and also, just as importantly, can be widely adopted so that it gives a broad range of clinicians and patients an opportunity to participate in studies that they wouldn't otherwise be able to participate in. So I take any questions.
So plenty of time for questions. I'll ask 1 or 2 to start off with. So Peter, I just checked on the NCI website, says there are 6,351 randomised phase 3 trials in cancer in the US. So over 6,000. How many registry trials would there be in cancer?
Good question. So
thank you.
Thank you. So this was I didn't mention it, but this is a concept that was first applied in cardiology. So in Europe, they conducted a number of registry studies based off their cardiology registries and studies of thousands of patients. It hasn't yet been adopted in oncology, so we're the first to do it. And I think we're in a unique position because of the strength of our registries. I think an important point is that you mentioned there's lots and lots of clinical trials underway. I'd suggest most of those are of new drugs and new drug combinations. And many of the important questions that need to be asked around sequencing biomarkers aren't being addressed in clinical trials. So for example, the circulating tumour DNA, the ECOG group in the US has been discussing a study in colorectal cancer for 10 years, haven't started yet. The studies are under discussion in the UK, but nobody's been able to actually get those happening. So it's working in a space that's important, a space that we've got strength, I think, and space that we can actually do things.
So let me just say that again. This is probably one of the first centres in the world, the VCCC, that are doing registry trials, if not the first centre, VCCC, doing registry trials in cancer at least.
Yeah.
World first.
Yes, I'm aware of cardiology and others, but this is just...
I'm impressed. Questions?
The registry, there seem to be a lot of different registries for different tumour types. So when you're going up to set up a registry, is there a common framework that gets used between them, or are they all completely set up independently, or how does it work?
Yeah, it's not straightforward to set up a registry, and over the last 15, 20 years, I've been around the world looking at lots of registries, and there's lots of failed efforts. So there's things you can learn from. It's a bit trial and error in terms of the data set that you try and capture. So you try and capture the minimum amount of data that you need, not every possible scenario. So that's an example about the data set. But with the registries that we've set up, we've tried to standardise across different tumour types, and they all look pretty similar. Hopefully they're all quite attractive, quite intuitive, and quite easy to use, because the worst thing is a registry that you try and enter data and you can't work out where you're going and how to navigate. So most of them are quite attractive, I think, quite easy to use, and that's critical to the success. But hopefully the brain registry, the pancreas registry, colorectal, all look pretty similar, and you can navigate across both of them. And we're going to learn from each of them to inform the other.
Questions? So use the microphone. Thank you. Introduce yourself to it.
I'm Vish, I'm an medical oncologist. I've worked with Mark and Peter for a long time, and I'm the Chief Medical Information Officer of PEDMAT. So, Peter, thanks for a great talk. So, I'm just wondering about a couple of things. So data quality in the registry, I assume, because you've got people that are, that's presumably one of the things being set up. Because even if we get an EMR, we're still stuck with the issue of how we can put in those fields that you've talked about, but it's really getting that data stewardship and people to actually care about entering that data. So I guess my question is about the data quality internally within your registries. But then more broadly, thinking about the international efforts where they do have EMRs, are people exporting data into registries, do you think, or is there any communication between EMRs and the registries, or is it a manual sort of two screens open at once and people copying the data?
Yeah, so the first question, in terms of data quality, it's a critical question. I think it's one of the reasons that you have a hard endpoint like overall survival. It's also critical that patients are allocated and take up the arm that they're randomised to. So that's one of the things we need to explore. And there's lots of aspects of these registry trials that we need to look at very carefully over the next 3 years, sort of in that feasibility phase, to understand what works and what doesn't work and what the hazards are. But I think trying to capture the data, you try and capture critical data elements, not so much adverse events, quality of life. Those things are more difficult to capture, but things that you can reliably capture and interpret. In terms of the efforts elsewhere, as you know, one of the challenges with EMRs is so many different types of EMRs, and they're often established, or they are established to manage patients, not to collect data. So the electronic medical record is not often a good place to go to look for data. And as far as I know, most efforts to extract data from EMRs have been quite unsuccessful. It mostly relates to the way that the databases are constructed.
Peter, can I ask, so the term turbocharge was in your title, and I know, you're driving the Ferrari colorectal turbocharge system, but most of us work in tumour streams that they're sort of DATS and 120Ys. How do we sort of get these things up to your speed? How do we get funded?
That's a good question. So one of the challenges when you're starting with something new is how to fund it. So if I put a grant into NHMRC for registry trials, probably not going to score that well. I don't have a track record. I haven't demonstrated the utility. So I think part of our focus is to work with the existing registries and demonstrate that we can do this, and that hopefully would increase enthusiasm and funding opportunities for more registries. But as a general statement, over time, there's more and more registries covering more and more tumour types. So now, when I'm talking about the registries we have for cancer, I'm talking about the ones that we don't have as a shorter list than the ones that we do have. So we have brain, colorectal. There's a few that are missing, and for various reasons. But over time, over the next 3 to 5 years, I expect we'll have a registry effort in every solid tumour type across the VCCC.
Is there an impediment? Is there pushback by people for registry, or is it territory, or what's the problem?
Yeah, I think the biggest issue is the data collection, having the resource to collect the data. So I'm conscientiously entering all the data on the patients I look after, but most of the clinicians are reluctant to do that. I think it's time and other aspects as well. So you do need to have good resources in terms of data management to collect the data to support the clinicians.
Other questions? Just wait for the microphone.
Yep, thanks. My name is Robin. I'm a German epidemiologist, a German professor in College School of Public Health. So I wonder, the first question about what we saw in Germany was a proliferation of registries pretty much last decade. So people kind of lost track, and we now do registries for registries, and it became kind of overwhelming. The second question is, are there any ideas to incentivize high quality data entry in registries, or do you rely specifically on like individual quality sense?
Yeah, on the first question, I think we've had a similar problem in Australia, and often you go to a new hospital and you find they've got their own registry, and within Melbourne there might be 4 or 5 colorectal registries or a number of brain tumour registries. Everyone's had the same idea at the same time. And one of the difficult things to navigate is getting a consensus that everyone is going to use the same registry. That's very important. There's now technology platforms like BioGrid that can link data from different registries. And as long as you've got standard data definitions, that's possible. But ideally have everyone in the same room working on the same registry to achieve this. And I think we've done that for some of the registries. So we've got a international efforts in pancreatic, colorectal and other tumour types that hopefully would support these efforts. In terms of incentive, I think the main incentive for the clinicians is hopefully to improve treatment for patients, but being able to be authors on papers included to improve their CV, I think, is a major motivation. But I think we do need to have the support around the clinicians to help with the data entry as well. So it is challenging to get engagement and to maintain the enthusiasm, but hopefully over time we can demonstrate the impact, and that's going to increase the enthusiasm for being involved.
All done? Okay, so I'll just, I think if I do that, we'll see what's forthcoming. So the next Monday lives are up there in small print, David Ball, I can see you squinting. Peter, as your ex-boss, I'm so thrilled you've done so well. Peter's been a quiet achiever and has achieved so much over a long period of time, working very hard, and admirable modesty, but has really been a remarkable quiet achiever. This is a token of our esteem. No expense spared. Actually, no expense, but thank you very much.
Thanks so much, mate.